Hello, good afternoon, everyone, and it's a pleasure to be here, my first ISMB, and it's been wonderful so far. So I think the first two talks, earlier ones, really segued into my presentation today. So I'll be talking of another related CRISPR molecule targeting, but it's not Cas9 and it's not targeting DNA, it's targeting single-stranded RNAs, and which is the Cas13, precisely the Cas13 BPSP ortholog. So myself, I work at the University of Melbourne, and I'm co-appointed between the Electrical Engineering and the Doherty Institute of Infection and Immunity. Some of you might know of that. So I'll just start with a sort of a broader context to my talk here. So I've been working for a number of years in the virus space and developing computational models, understanding viruses, RNA viruses, and trying to help identify hotspots for vaccine design and so on. So what led me to this? Giving this context. So from the WHO last year, I think they had released this priority pathogen list of looking at the possible viruses and bacteria which could be part of the next pandemic, and there's a lot of movement around pandemic preparedness. So looking at this list, so there are 11 viral families identified by WHO, where they've identified specific viruses and recommended to really focus on these for developing therapeutics and vaccines. And interestingly, a large fraction of those, we do not have any vaccines or any antivirals against them. And even more interesting from the perspective of today's talk would be, out of these 11 families, 10 of them are RNA viruses. So the point over here I'm trying to get at is we really need new and very programmable platform technologies for developing both vaccines and antiviral therapies against RNA viruses. So, okay, the last figure on the right is also trying to get across that, because there's this, we have seen with SARS-CoV-1 back in 2003 and SARS-CoV-2 is fresh in our memory. So we see that there's a reason to really target not only very specific viruses. That's what the development has been broadly so far. We need to even look broadly at viral families and subfamilies. So that brings to the context here. So now very specifically about the RNA therapeutic design. So apart from the challenges of looking at family of viruses, there are more challenges is looking at the mutation rate, which, as most of us know, that most of the RNA viruses have a very high rate of mutation, and also under effects of immune pressure and selective fitnesses, they can also generate a lot of variants and quasi-species. A classic example is HIV, that after decades of work, we have not yet designed a vaccine against HIV because it has a crazy rate of mutations and replication. So the point to really take away is that we need reprogrammable and platform technologies for designing antivirals. So, okay, now for targeting RNA, we have, there's a lot of different technologies. One of them very popular has been the RNAi, RNA interference, but it has its limitations because of some, again, off-target effects, and it's really difficult to model over there. And also it's very lax in its targeting. Specificity is a challenge over there. So now what that brings to the main focus of today's talk. So that would be on Cas13. So Cas13 has been discovered less than 10 years ago. So it's a fairly recent mode which people have discovered from bacteria. So one of the really promising features is it has a very large spacer sequence. So we have talked about the 20-length spacer sequence in Cas9, but Cas13 offers up to a length of 30 nucleotide. So hence it could potentially be very specific. And also, I mean, generally, I'll be talking with the respect of virus targeting, but more broadly, if we develop this technology, we can really even leverage it for targeting oncogenic genes and so forth. And one of the advantages is that because it doesn't target the DNA, so yeah, it doesn't have that a bit of a safer modality as compared to targeting DNAs. Right. So as most of us, I think, know that, so this has developed in different species, Previtola, for example, the PSP one. And I think I'll just move through this quickly. So there's a guide RNA, which is loaded, and then once it's targeting, the only difference from the earlier talk, it's targeting a single-stranded RNA. Otherwise, it's pretty much the same, and that's what gives us the idea that this targeting and degrading of the target transcript can potentially be used as an antiviral strategy, and if it works, it would be wonderful. So what brought me to this is one of our collaborators back in Tori. They did this study at the end of the pandemic, and they did find that we can, as a proof of concept, basically target a SARS-CoV-2 transcript in a cell line, and it can potentially be useful. So, but they had a lot of limitations, but at least they showed this as a proof of concept. So now that brought a lot of interest, and the question then said, do we have design principles? Because if you're thinking of taking this to the next step and use it as an antiviral strategy, we need design principles. We need to understand what are its off targets and on targets and other activities, which are generally very poorly characterized because it's an understudied molecule. So among the challenges, it's like people generally in the lab have a very limited data set of using trial and error. They can find some guides which work, some they don't, and generally this is a very inefficient process. It takes a lot of time and resources to generate the data in the laboratory, and definitely not feasible for upscaling. So now, and also potentially we can see, because of the large spacer length, there are large number of guides to test if we really want to model. So now that's where we came into the picture, and I'm just trying to get across another point today is like being involved very closely upfront at the level of designing the experiments with the laboratory researchers really is something which we usually don't do. I think it's rather uncommon that people from the computational side really step up and get invested in the biological side of things and in the design phase. So this has been a very refreshing experience for me. So we were involved very upfront, and then we said, okay, we even need to take a step back and design a reporter system, because one of the challenges is that people reporting efficiencies of CRISPRs from different laboratories, it's difficult to really reproduce them. And even like, for example, we found guides, the same guide sequence from one laboratory, one experiment, reporting a target efficiency of 20, 25, the same guide reporting a targeting efficiency of 70 and 80. So the normalization and all those effects really play a lot of role. So then we just sat with our collaborators and we picked up an mCherry RNA. So very briefly, it lights up red. And if it's being targeted, we don't see that red fluorescence. So then we said, okay, let's design a very systematic way of targeting this mCherry in the same laboratory setting and generate our own data from which we can try to learn. And then we did that in the head cells. So we selected different transcripts, but mCherry is the one I'll be talking about today. And then we tried to say, okay, if the length is around 30 nucleotides, so let's have a very comprehensive screening, like a single tile shift across a single position. So that will give you around 60 to 61 guides to test. Now, this is the figure where I like to spend a bit of time. So over here, we can see that the bars, so the y-axis is a normalized relative expression. So if it's high, if it's not really targeting. And if it's 0, it means that the transcript has been targeted and we don't see any fluorescence. So one of the regions of interest would be if we see around here in this position, we can see even with a single shift, for example, 38 is like very good targeting. But even with a single shift of a nucleotide, we see that the guide number 39 is like there's almost no targeting. So this is really very interesting because something in the field people generally, it's not known for Cas13b, first of all, but generally people don't really pay a lot of specific details on the target composition itself. But this came out to be a very, very interesting outcome that, well, the target sequence itself seems to play a lot of role. Another, I'll just mention quickly, is another thing is because we are doing it in a very systematic controlled environment. So we try to really control for a lot of other factors like the loading efficiency of the RNA within the Cas13 groove, and also the contextual association of them. So I mean, it really shows that it's highly likely that it's just the guide sequence which is influencing this significant change in the targeting efficiency. So move forward, we did a very simple computational work. We looked at the distribution of the guides. We found the ones which are pretty good, maybe above 70-80% targeting efficiency, and we found a set of guides which are poor. And we did a very simple computational comparison between them, just using weighted matrices and enrichment plots, and just doing the delta frequency plots between them. So what we really found, so the first message was that we really find that there is sequence-specific recognition. which I just mentioned, and then also position dependent. So we did a lot of like shifting of these guides, and we find that having like the identity of the nucleotide at a specific position is also associated with the targeting efficiency. So yeah, so what I mean, the main takeaway previous to this, people had that maybe the sequence motive is not that important, and it was just like if you want to target any transcript, you just need perfect base pairing in the Cas13, but this really showed that it's not true. We also looked at some other potential determinants of cRNA efficiency. Over here, for example, the spatial, the folding minimum free energy of the folding RNA or the guide, and the interaction energy and so forth. We didn't really pick up anything. The main signatures we picked up were the content of GA and C. So it seems like for this Cas13b ortholog, enrichment of G in the spacer guide seems to have a positive correlation, whereas enrichment of C seems to really negatively correlate. And then we did some basic linear regression, positive, and other very basic simple learning tools. So the key message which came across was that at the 5' end, that would be the start of the guide, an enrichment of G really associates very well with an increased and very specific targeting efficiency. So yeah, so I'll just, so we also did some, then we guided some of the follow-up experiments. Like for example, if you take a guide which has a very good efficiency, which starts with 2 Gs at the 5' end, what if we just change them with Cs? And as these figures are telling us that just replacing the first 2 guides in some context can really abrogate the efficiency almost entirely. So that seems to be a very basic, but a very, very powerful biomarker. Just starting with 2 Gs can be good. So then we also looked at mismatch tolerances. So another thing, okay, how many mismatches can these guides tolerate if you want to really go ahead and design an antiviral target? So we looked at all sorts of different combinations, its starting position, ending position, and sliding in between, and so on. So the takeaway was that generally at the 5' end, mismatches are not well tolerated, but on the 3 and up to 3 nucleotides elsewhere, the guides generally they tolerate. Right, so we also looked at one of the key issues, as we've heard earlier, is also the collateral activity. So we designed some experiments with the collaborators looking at getting at the M-cherry as the target and also having GFP as a collateral. And we did report it for the Cas13 for the first time that this ortholog of Cas13b has a very minimal collateral activity. And that's another thing which really adds really a strong reason to really pursue this one further and try to use it for downstream applications. So I think I'll have to move a bit fast here. Right. So now what the point was, okay, another point would be like we also looked at the off targets in the laboratory. I won't go in the experiments of that, but in the human transcriptome. So we did look like, for example, in the just loading the Cas13b without the guide, we didn't see any downregulation of a large number of proteins. But in the presence of the PSP Cas13b with the guide, so the 2 targeted transcripts, the BCR ALB1 and the EGFP, they had significantly high downregulation. So what this is telling us that within the context of the HAC cell lines, human cell lines 293T, it's a very limited, I mean, we don't see any significant evidence of collateral with other expressions as well. So all in all, so these were all very promising. We just thought, okay, we need to do a lot of follow-up on this, but we can come up with a very simple design tool as well. So we just, we had this where we developed this very simple, straightforward web interface where you can give any sequence of RNA and or DNA, and it can give you different suggested guides with different potency scores, and also screens across the human transcriptome for any potential off targets. So this work is, I mean, this is what then we had the outcome of the work was published as a paper. So what was really very interesting, as the start of the talk, I mentioned that there's a motivation that why we're looking at this. So we really wanted to take this, even a very simple sort of computational knowledge, and how we can integrate it with the downstream applications. So we were lucky to have further collaborators, and one of them tested on some oncogenic RNA genes, and the other one, which I'm very excited about, is looking at the HBV, hepatitis B virus. and that's also an RNA virus, and it's a huge challenge. We don't have any effective drugs against it. So what they did was they used our system and to identify some of the guides for the E antigen for HSB, and they took it to the next level. They basically used the AAV as a vector to deliver the Cas and the guides in the mice, and as a first proof of principle, they did show that it is able to very effectively suppress replication within the mice itself. So this actually laid a lot of stage, and we have some ongoing further collaborations on this. So I'll like to really summarize now. So for the first time, this study elucidated that there are key molecular design principles. So in the whole scheme of designing or taking Cas13b as an antiviral modality, we really need to take the sequence into account as well. The target sequence, which also brings me back to the point that if you're going to take this to target broad families of viruses, then there's a lot of challenge with sequence diversity, immunescape mutations, and so on. So this then really opens up the question of we really need to find the robust targets, and we have a lot of work in progress on that as well. So yeah, so the just last three. So what are the next challenges is we did a very simple computational learning. So we are, as we speak, we are generating very high throughput high throughput data for Cas13b and also some of the other related orthologs. We'd really like to take it to the next level and use more sophisticated machine learning algorithms, for which we need definitely more data. But again, I really want to stress this point that generating it within a very controlled experimental design, even if we don't get thousands of guides, even hundreds would be fine if they are all within the same experimental setting, so that we can really compare the fluorescence and the efficiencies which we are getting, as opposed to gathering data from different labs, which we find a lot of inconsistencies. And and also which is beyond our scope, but also some optimization with the in vivo delivery mechanism. I didn't go into the details, but there are some factors from the vector which we use to deliver the CRISPR and the guide RNA that influences the efficiency as well. So that also becomes as a factor. So this is again coming back to the point that being very closely involved with the experimental partners, so that if we are sitting with them when they're designing their experiments, so then we can really inform them, and their limitations can really inform our design of computational learning tools. Right. So with that, I think this is like where we would like to really go, is that if we want to target a family of viruses, for example, coronavirus family or flavivirus or Nipah we have a pipeline. We can really quickly upscale and test them in vitro systems, and hopefully then test them in live viruses as well. So we already have ongoing collaboration. It's unpublished, but we have had a success with Nipah virus targeting as well in BSL-4, and we're really excited. It's still not published, so yeah. So at the end, I'd like to really acknowledge all the funders from mRNA Victoria, ARC, and also shout out to iRNA for the conference fellowship, and my McKinsey postdoctoral fellowship, and our collaborators from Peter Mac, Doherty, and the Cummings Center. Thank you.
Very nice talk. I was wondering, so previously the 3 and downstream of target, there's a PFS domain, which is a non-G target. When I see your plot, it seems downstream have more hit as kind of importance.
So the PFS, so okay, that's a very interesting observation. So I'll, so this one, so over here, so what we also identified from the data set that this ortholog of Cas13 is not really affected by the downstream PFS sequence. So the other orthologs, that the RFX13D has a strong PFS influence, but PSB Cas13D seems to be, and that actually gives us this real, it expands the potential target space.
Hi, I have quite a biological question, so it may be a bit far from the talk that we just saw, which was great, by the way. We work with Cas13D, and one thing that we see is that because it cleaves the RNA, depending on where you cleave, you can still see biological activity, which is different from the normal biological activity that you would expect, but it doesn't obliterate it because you just cut the RNA. The 2 different chunks that you create can still be active. I was just curious if you saw any of this or any indication of this in your work.
Right. So okay, so it's Okay, I can answer to what I, to the extent I know about it, is so for the biological activity, if you're referring to the expression of the downstream, okay. All right, so what we have seen, so again, our primary goal is to look at viral transcripts. So these viral transcripts are they're very sensitive to targeting, and we do have, we do have some constraints, like we don't want to really target the UTR 5' or 3' downstream UTR regions. We are normally targeting the CDS regions, so which so far we haven't seen any. Once it's cleaved, because it's a subgenomic RNA, it really doesn't show any downstream expression. So I hope that answers.
Okay, if there are no further questions, then thank you for a great talk, and let's thank all the speakers for a great session this afternoon.